Hello, everyone, and thank you so much for joining us. My name is Nora Romero, and it is a real pleasure for me to be hosting today's talk as part of the Biogeography and Systematic Talks. This webinar series has been organized by the Packard Lab and the Center for Ecology, Evolution, and Conservation at York University. So just a couple of reminders before we start the webinar. As usual, after the presentation, we will have a Q&A period. Please, we ask that you include your questions into the Q&A feature that you can see at the bottom of your screen. This will make sure that your questions don't get lost with all the chat entries. Of course, all the other chat entries are welcome. If the discussion extends past the hour mark and you can no longer be present, do not worry, we have you covered. Both the presentation and the discussion are being recorded. However, we will have to wait a little bit until we can release the presentation, as the author has requested that he will publish the results before making the video public. But of course, we will be notifying you when that happens so that you can go on the YouTube link and enjoy the video at your convenience. Now, before we begin, we would like to acknowledge the land that we team at York University are coming from. We recognize that many indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. The university acknowledged its presence on the traditional territory of many indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many indigenous peoples. We acknowledge the current treaty holders, that is, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Agreement to peaceable share and care for the Great Lakes region. If you would like to know more about the history of the land that you come from, or the history of the land which you're joining us from today, this is a fantastic resource for this exploration. www.native-land.ca. It started as a Canadian initiative, but now it has a worldwide coverage. And now I would like to welcome Dr. Lawrence Packard, distinguished research professor at York University, will greet you and who will lead the Q&A session. Go ahead, Lawrence. The stage is all yours for now.
Thank you very much, Nora, for that introduction. So it gives me great pleasure today to introduce Eduardo Almeida, who I've known for quite some time. He did his undergrad in 2000 and master's in 2002 under Fernando Silveira's supervision at the Universidad Federal de Minas Gerais. From 2002 to 2007, he did his PhD in entomology at Cornell under Brian Danforth. In 2006, he taught me how to say estupidamente gelado, which is what happens when you put ice cream that is so cold into your mouth that it gives you a headache. From 2007 to 2009, he went back to Brazil as a postdoc in Gabriel Mello's lab. 2009, for 2 years, he got his first job as a professor, as an assistant professor at the Universidad Federal do ABC, which is an interesting name. 2011 to president. He's been at the Universidad de Sao Paulo, Ribeirão Preto campus, and his ongoing research is about bee biogeography, which is the subject of his evolution of biogeography, which is the subject of his talk today. So welcome, everybody, and welcome, Eduardo.
Thank you, Lawrence. I think I can turn on my video. I think I need Nora's assistance for that. All right. So I'm going to share my screen. And I hope you can see me all right. Can you see me, Lawrence?
Yeah.
Nora?
Yeah.
Okay, and you can see the presentation, right?
Yeah.
Okay. Okay. So thanks very much for the introduction. I more or less knew what Lawrence was going to say, but it's unavoidable to give some laughs when we hear Lawrence saying that, especially because of the Portuguese and all the challenges that that impose. But I think the main messages were well conveyed. So thank you, Lawrence. And before beginning to talk about the actual subject of this presentation, I would like to thank two people here. One is Silas Bossert, who you probably, most of people following this seminar series have seen a couple of weeks ago, talking about andrenids. And not only Silas has been this amazing growing scientist in bee research doing very exciting work, but he's also part of the results I'm going to show here today to you. And there are other co-authors, but Silas has been an amazing partner in making everything work. And I'm going to introduce everyone else in the end of this presentation. And the other one is, of course, Dr. Packer. Lawrence is the one on the left on this right-hand picture you see. And Lawrence has been part of much of what has been going on in my research for a number of years, doing field work together, discussing phylogeny of colletid bees, providing samples, and more recently inviting me to be part of this seminar series, which is a pleasure. So thank you very much, and congratulations to the Packer Lab and all institutions involved in making this seminar series possible, which is really awesome. I would rather be in Toronto doing this locally, but it's good to be in touch with bee researchers and bee lovers around the world. So this is the title of the presentation I kind of thought about bringing here to you today. So it's integrating phylogenomics and fossil. There is something that's covering. I can't see everything that's in my screen, and the fossil record to interpret and the fossil evidence to interpret early bee biogeography. This sounds challenging, and it has been. The idea sounded simple when it started about 7 years ago, and every year has been increasingly more challenging. And but we're getting there. Of course, as I mentioned, with the assistance of a number of people, especially Silas, we're getting there, and hopefully this is the spoiler of what's coming next as a published paper, which we expect to submit in the next couple of months. And the main topic here is bee biogeography. And I don't think I need to define what bees are to you. I think if you decided to follow this seminar series, you know bees quite well. So I'm not going to go into the trouble of telling what bees are. But not everyone is so familiarized with biogeography. And if you need to learn biogeography, and if you need to learn about the history of bee biogeography, this is the essential paper to go to. This is a monographic work published by Charles Michener in 43 years ago, 1979, Biogeography of the Bees. I think the title says everything. It's broad in scope, and Michener revised everything that was known at that point in terms of bee diversity and how it was distributed around the world, and how it came to be distributed the way it is today. And it's really an amazing piece of work. And about that time, biogeography was a hot topic, as it still is. And a year earlier, Platnick and Nelson published a very famous paper in which they defined biogeography as the science that will try to answer this question: why taxa are distributed where they are today? So we look at current distribution and we try to find answers for that. And that's more or less what Michener did in a very clever way, as it was most of, or almost everything that Michener did during his academic life. Michener said that in some sentences, I think that they are important for those who have not read this paper. He talked about how the distribution of bee richness happens in a general pattern, recognizing that warm temperate areas, especially the xeric ones, are particularly important to study bees, and they may tell us about the origin of bees. They may preserve some of the archaic groups of bees. And based on that, and based on fossil evidence that was known at that point, and paleo reconstructions and a number of other factors, they were integrated by Michener to propose that most likely bees originated in Western Gondwanaland, so the areas that now correspond to Africa and South America, sometime during the Cretaceous. And that's quite amazing, because that was before we had all this fancy ways of estimating the time of origin of taxa. We didn't know as many fossil taxa as we know today, and many of the tools that make our life much easier in some ways to answer these big questions. And in fact, if we fast forward to 2021, this nice paper that most people have probably seen was published by Michael Orr and co-authors in 2021, Current Biology, in which they investigated global patterns and drivers of bee diversification or bees diversity. And just to give you an example of one of the main results they got, we see that this prediction that bee diversity is going to be higher in following this bimodal latitudinal gradient makes sense. So it's in many ways a prediction that Michener made in 79, but it was refined and investigated in much more detail by Orr et al. So that's, I think it's also a lesson of how scientific knowledge increases through time. Sometimes we have great hypotheses that given a number of decades and a number of clever people who dedicate themselves to analyze those hypotheses, they conclude it's right or wrong. And this is one of these cases in which we say, oh, Michener got it right, which is mostly the case. And Michener was very humble too. He thought that, and he wrote that in 79, that it may be premature for someone to investigate what went on with the history of bees, because at that point, they were just starting to use cladistic tools to investigate phylogenetic relationships. He was aware of that, but he said, okay, but I think it's a good time to at least summarize what we know. And what happened next? We learned a lot about the trees that depict the phylogenetic relationships among bees, right? We spent more than 40 years planting trees of bees. And that was based on improved knowledge on species level diversity and distribution. So we've described since the late 70s, thousands of new species, and now we estimate to know about 20,000 species of bees. We have developed numeric methods and ways to code characters to improve our understanding of phylogeny of whatever taxa, but of course we're interested in the most interesting ones, bees, and that was done for bees too. And we had this what could be called a phylogenetic revolution in the 90s, which went on through the 21st century. And we have morphology-based hypotheses and molecular-based hypotheses for most bee lineages, which also allowed us to have classifications that reflect this ever-increasing understanding of bee phylogeny. And of course, lots of fossils were added to our understanding of the fossil record of bees, which is, of course, very important to understand this evolution through time. For those who don't know much about Michener, may have heard his name, I really recommend that you learn, you go on and look for sources to understand what this amazing scientist did for bee research and science in general. So in 2007, he published sort of an autobiography in the Annual Review of Entomology, which is extremely interesting. And Michener left us in 2015, and a number of papers published about his life and career are incredibly interesting and depicting and depict very well what he has done. Especially, I should recommend Michael Engel's 2015 paper published in the Journal of Melitology. And he had this outstanding role in describing and interpreting what's going on in terms of bee diversity in a world level. And I love this picture in particular, showing Charles Michener and Byron Alexander, 1994. And I generally say that they look happy, and they should be happy. That is, 1994 is the year prior to the publication of a famous paper, Alexander and Michener, 1995, which came out 2 years after Roy Gelsine and Michener, 1993. And these two papers, they revolutionized bee systematics, because they studied bee systematics in a comprehensive way based on very large morphology-based data sets. And they have been fundamental to delimit the groups of bees that we now see in our classifications, to refine a number of complicated matters in terms of bee classification and understanding of their relationships. And that was part of the contribution made in the early 90s, which went on in the years to come. And here I show two very important and impactful papers published more recently, one in 2013 by Sophie Cardinal and Brian Danforth, and based on molecular data of traditional methods of DNA sequencing, or Sanger-based methods, and more recently by Michael Brandstetter and co-authors in 2017, based on a phylogenomic data set based on next generation sequencing. I'm not going to talk in detail about what these amazing papers have provided us with, but I'm going to mention that more and more discoveries made before the 90s in papers like Roy Gelsine and Michener, Alexander and Michener in the early 90s, and so on. They help paving the way for ever-growing refinement and confidence on the relationships of bees, which groups should be recognized as monophyletic, and even the ages of these groups, as I'm going to talk more in a moment. And to try to expand this and to understand more about some of the problems that still remain, I've been involved with two initiatives, one using molecular data, here shown in a way that's going to be almost impossible for anyone to interpret what's depicted in this tree, but it's a molecular-based data set with UCE data, this ultra-conserved elements. So it would be classified as a phylogenomic analysis of bees, which sampled 216 species, representing all families, subfamilies, and most tribes. And then it's going to show more or less the same as we've seen before in previous studies, really consolidating some of this understanding we've been building over the last decades, but also improving the placement of some taxa. And then another one based on 116 species of bees sampled based on morphological data. And that was the most important part of the PhD thesis of a former student of mine, Diego Porto. And here again, it's a large data set with almost 400 morphological characters from internal and external skeletal morphology. And again, we see congruence between the results we get here and results that we've been getting with different kinds of sources of data and different kinds of analysis. And as take-home messages of what's going on in terms of this learning process about bee phylogenetics, we can say that the more than 20,000 known species of bees, they fit well within the classifications we have, which in turn reflect the phylogenetic relationships that we've been learning more and more in the case of bees. We can also say that congruence among these different hypotheses, they make our confidence on the right tree to grow. We're never going to know if it's the right tree, but if we're thinking about confirmation in terms of scientific progress, I think we're getting closer to the truth. That's what we hope, and that's what drives us to keep doing science, right? But also we can say that there are some hard or very hard problems in terms of phylogeny to be solved. And that's why it's still advisable for us to keep doing our systematic research, because that's going to be very important to solve some of these outstanding problems that we have. So I go back to the title of this talk, and I've talked about phylogenomics. I said just a little bit about the fossil evidence, but I should turn my attention now to the biogeography, and that's what I propose myself to talk to you about today. And I gave you this question, right, that defines biogeography: why taxa are distributed where they are today. But I very much like another question that can be asked to define what biogeography is about, which is, why is diversity unevenly distributed across Earth? So if we look at these maps that show the distribution of species richness by family in the different biogeographic regions of the world, we see that andrenids, they have a higher species richness in northern continents. We see that it's very high, as marked by the hot colors, in the Palearctic and in the Nearctic, more so than in the Afrotropics, for example, and much more than in Australia, because andrenids are not in Australia, so they're absent from there. Or in case of apidae, we see a lot of apids in South America, and we see a lot of apids in the Palearctic. Of course, it doesn't mean that the same lineages are represented in one part of the world and the other, but we still have very high numbers of species. Or Colletidae and Cynotrididae, which are very diverse in Australia and have different levels of diversity in different parts of the world. What does it mean? Does it mean that if we have high diversity, we have a probable area where these groups of bees first diversified, where they originated in the sense that the lineage diverged from its sister group? What does it mean? Or it's just the outcome of a very complex process that may indicate a place of origin or a more recent set of events. And that's what biogeography is about. We have to look at different stages in the history of the taxa we're interested in, and try to understand what were the events responsible for resulting in this or producing the kind of outcome we have in terms of distribution. And of course, we have to think about time. So I showed you a highlight of this important paper published in 2013 by Cardinal and Danforth, in which, for the first time, someone showed a time tree or a time-calibrated tree and a temporal framework for diversification of the diversification of bees as a whole. And so these authors, they had all major lineages of bees represented in their sampling, and they have a time scale in the bottom, and they show when the different clades, they diverge from their close relatives, when different subgroups started diversifying, and they learn a number of important lessons, among them the one that makes the title of this paper, bees diversified in the age of eudicots. So they had this estimate for the origin of bees. So between 110, 130 million years ago, which is the early Cretaceous, the same time when eudicots, or the major lineages of flowering plants, also diversified, which is in a way expected, right? And it had been proposed by authors before Cardinal and Danforth, but it's very cool to see an analytical result that's going to match predictions that were based on a good sense of what makes sense in terms of evolution. And so it's clear to us that bees, because they're dependent on flowering plants, they couldn't have diversified prior to the food sources they seem to rely on. And it would be strange for all the flowering plants to have diversified before bees arrived, and then they just took advantage of this great diversity. So there was some kind of synchronicity, which Cardinal and Danforth demonstrated quite nicely in this 2013 paper. And to take advantage of a time-calibrated tree and to know the time frame of evolution is extremely important for almost any question in evolutionary comparative biology, including, of course, biogeography, which will look at the space, that's the geographic part, but also look at the time frame for the changes to happen. So what we really would like to have, and I certainly thought about the time machine like this, is to have some kind of device that would allow me to go back to this time when bees were inferred to have originated, so 120 million years ago, and travel around the globe and look for bees in different parts of Gondwana, in different parts of the northern continents, pardon me, and see, okay, here's where I find bees million years later, 110 million years ago, and look for bees. Where are they? Are they still in the same place where I found them 10 million years earlier, or they have changed their location? Do I have more kinds of bees? Which lineages in comparison to the present day lineages are represented, and where they are they in different continents, in different areas of the globe? And then keep doing that from time to time. Maybe every 10 million years I could go back and to the planet and travel around to see what I could find. And I'm sure you all know that's not possible, still not possible, and we're not going to be able to use this time machine. And the time machine we have access to is the set of analytical tools based on phylogenetic based tools, the fossil record, and the distribution of current day diversity of bees. And we can try to integrate everything and make something that resembles a true time machine, but it's not going to be as perfect as it is, and see what we get from this attempt. If we think about the fossil record alone, and that's the picture of the planet Earth 120 million years ago, that's the fossil record we get for bees, for insects that are confidently described as bees. We don't see anything. And it's not a problem with the screen of the computer you have in front of you. It's a problem of the fossil record. There are no records of bees or insects we can confidently say this is a bee 120 million years ago. So we have pretty much one of two options. Either our estimates of bees being as old as 120 million years is wrong, and it's okay not to have bees around 120 million years ago, or the fossil record is incomplete, as we know it is, and we just don't have the fossils that will allow us to understand where bees were so long ago as in this case. For people working with birds and dinosaurs and stuff like that, they have a good picture. They almost can recreate a time machine. And if someone says, how did the world look like in the Jurassic? Let's go back 145 million years ago and look like and look for life and see how it's distributed. And then you see this this picture, which may be a very realistic picture of the world back then. Some people get enthusiastic about this. I find it boring. It's boring because first, I don't see bees, and I challenge someone to find a single bee there. And I don't expect to find bees because, in fact, you see this strange plants. They look like conifers. They don't have flowers. So it's very unlikely there were bees flying around. Even the Cretaceous, if you look at this, some people may find it beautiful to have this big, awkward lizard in the middle, but all around, all I see are conifers. And so it doesn't look like a very promising habitat for bee research. This is much better. This is more towards late Cretaceous. We still have a big lizard like posing as a very important animal in the middle. We don't see the bees. But if I had a drone, or I could look around and find some flowers, because I see flowering plants in there, I would probably find some bees. But that's not part of the paleo reconstructions we see made by paleontologists. They generally don't pay much attention to bees. And of course, it becomes really promising when we move forward to the Cenozoic, and then we have a lot of flowering plants, and we have these other less interesting animals, but we certainly have lots of bees flying around. So the fact that we don't have a good fossil record for bees in the early stages of what we presume the early evolution of bees to be, it means that they're not useful. Well, of course, the fossil record is useful, but it's not enough. It's going to be enough to answer a number of questions, including some I'm going to show to you in a moment. But this is a good example of a paper that is that makes very good use of the fossil record. This classic paper by Michael Engel, published in 2001, shows how current diversity of corbiculate bees and extinct diversity based on a very rich fossil record from the Eocene shows that a number of lineages went extinct towards the end of the Eocene, when the planet changed radically, and a number of other taxa also went extinct. So thinking about about half of the, if we think about 120 million years ago as the beginning of bee evolution, and from there to the present, and we're in the middle, 60 to 45, 60 to 30, we have a lot of fossils that are extremely important to make us understand with a great level of detail what went on with part of the bee evolution. And in this research, we try to take advantage of what's known from the fossil record, even if it's restricted to some temporal layers through bee evolution. And now talking about some of the papers that were fundamental to improve our knowledge about groups of bees, but not bees as a whole. I'm going to talk to show you and remind you of some important landmarks in the study of bee biogeography, and maybe introduce to some of you some of these papers. The first one was published in the early 2000s, and by Brian Danforth and co-authors, 2004, in which they studied collected bees. And this is a very important paper because it used two sources of data that became prevalent in biogeographic analysis. a time-calibrated tree, and an event-based method, an explicit event-based method used to do the analytical for the analytical approach to study bee by historical biogeography. So that was dispersal vicariance analysis, a DIVA analysis. And in this paper, Danforth et al. Found good signal of vicariance and association to southern continents when they studied the earliest divergent lineages of collected bees. Moving forward to the present, this year we had 2 papers published about Andrenidae, and the first one by Bossert et al. And the second one by Ramos et al. And in both cases, they were able to find out that Andrenid bees had their early diversification in the late Cretaceous, and mostly happened, most of the important biogeographic diversification events taking place in South America, with later expansions of their ranges to North America and to the Old World. It's also interesting that most of this 3,000 species, they derive from quite recent diversification events happened in the last 15 million years, with a very high diversification rate happening in recent times, especially in 2 genera, Andrena and Perdita. And based on this kind of analysis by Danforth et al., by Ramos et al., and Bossert et al., we can really get a good picture of what kinds of events and when those events took place to shape current bee biogeography of this specific taxa. I shouldn't avoid, or I couldn't prevent myself from talking about Colletidae. This has been a major interest I've had for the last 15 years, more or less. And so in 2012, my co-authors and I published this paper, 2012, as I mentioned, about the general understanding of bee biogeography, of colletid bee biogeography, I'm sorry. And what's interesting about Colletidae is that it has this very peculiar characteristic lineages associated to southern continents. So to Australia, to South America, and to Southern Africa. And that was thought by some people as a very classic example of Gondwanan association. So a taxon that would have arisen before the breakup of Gondwanaland, and its provincialism with taxa only present in some of these major southern areas could have been resulted from this early geological changes in the planet. But in fact, what we found out is that the most ancient divergences happened in Colletidae didn't take place that long ago. It was after the breakup of Africa and South America, but a persistent connection between Australia, Antarctica, and South America was very important for interchanges to happen between these continents and to create this historical connection between the Australian and the South American fauna that we see today, including, of course, Stenotrididae as well, which is the closely related cousin of Colletidae and endemic to Australia. And the last one is I want to mention today is the study by Littman et al. With Megachilidae, published in 2011. And in this research, the authors found a strong signal for vicariance events that would have shaped the initial divergences within Megachilidae, with endemic taxa placed as the earliest diverging lineages in either South America, Africa, or other parts of the Old World, really indicated, based on the biogeographic analysis and the timing of these divergences, to have happened before the complete separation between Africa and South America. So between what Michener and others have said about the association between the early diversification of bees with southern continents, and with what recently published papers on the biogeographic analysis of different bee families, we really find this indication of a close association to Gondwana and events that took place in the southern part of the planet. And this is an important study that tried to really answer, so can we point to Gondwana as the initial setting for bee biogeography? So in 2013, Hedke and co-authors published this impressive analysis of more than 1,300 species of bees and apoid wasps sampled for 20 genes in a supermatrix approach. And that covered 2/3 of the bee genera, which is really impressive. And try to analyze the history of bees in a very comprehensive way. And they found for each one of the families, as you see in the cladogram on the right, you see for many of the families signals that involving the historical signal involving southern continents, so SAM, South America. You see in some parts Africa, like close to the Colletidae, but you also see a number of little squares that have a relatively low or an average probability of being the most likely reconstruction. So there is some level of ambiguity in the results by Hedke et al. And there is a lot of doubt in terms of the reconstruction we can have for the root of bees. So what is the most likely setting for, or geographic setting for the origin of bees and the early diversification of bees? And in this study, due to, I would say, computational limitations, they didn't do a time-calibrated tree. And that reduces also the number of conclusions or the kind of analysis we can do in terms of biogeography for bees. So in this study, after this very long introduction and context for why should we do a biogeographic analysis of bees, we try to integrate 3 or 4 components that would hopefully provide us with an up-to-date, decisive as possible and comprehensive result for bee biogeography. So we have this phylogenomic framework with extensive taxon sampling, which of course takes advantage of previous research on bee phylogenetics to know what to sample. We have a divergence time estimation for this taxa we investigated. We're going to do a model-based biogeographic reconstruction for bees, and we're going to have that understood also in terms of the paleobiogeography based on the fossil record of bees. And this is a paper that's in preparation, being led by Silas Bossert, myself, and a number of co-authors. And this has one shortcoming, as we learned during the execution of this research. We're going to have the most promising results, or the most reliable results, relative to the early bee diversification portion of the history of bees. And our confidence on the results becomes lower and lower as we approach the present. And that's why it's important to look at the results I'm going to show you. At the same time, we look at this family-based level studies or subfamily-based level studies for groups that are going to be investigated in more detail by other authors. So, as I briefly mentioned before, we have a tree that has that includes 216 bee species That's 1% of the known bee diversity. That's not a lot, but we're pretty confident that we don't have any major lineage that's not represented. So all 7 families that are in there, and we have a good representation of taxa that are endemic to the relevant regions of the world. And you see these little squares close to the nodes. So those are support values, which is hard to interpret when we're talking about phylogenomics, because the support values tend to be very high. So what we did was to try to disturb the tree building as much as possible, trying to use different analytical methods and changing the way we sample the data. And in most cases, we can disturb the quality of the data as much as we want, but the phylogenetic signal is very strong, and it's in agreement with most previous studies, which gives us confidence that we're closer and closer to having a stable tree. But in some cases, we still have hard phylogenomic or phylogenetic problems, which need to be tackled in more detail in future studies. With this divergence time estimation, so including all the sampling of bee species, and 185 fossils analyzed together with it in a fossilized birth and death estimation in a Bayesian framework. And that's the merits of these analyses are all by Silas Bossert, has made an amazing work in terms of how to apply a fossilized birth and death approach to divergence time estimation, as done in the unbranded paper recently published. And again, we used various strategies to create subset of the loci and make the data set computationally tractable to be analyzed. And in the tree on the right, what you see is a comparison of the results we got in the present study and previously estimated ages for major bee groups, so bee families, and clades or superfamilial clades of bees, like the short-tongued bees, the bees as a whole, long-tongued bees, and parts of the short-tongued bees. And of course, there is variation, but in general, again, we see that the congruence among these results gives us a lot of confidence in terms of how close we're getting to results that make sense and will hopefully not change a lot in future research with the phylogeny of bees. We can talk about biogeographic analysis as backward analysis. So if you never thought about how a biogeographic analysis is done, it's a backward approach in which we think about the distributions of taxa in the present, and then we use the tree to move backwards in time to the origin of the clade or to the origin of the whole lineage of interest. And for that, we used a dispersal extinction cladogenesis model, or DEC model, with the recognition of 6 big areas, which are shown in the bottom left. We used, of course, the time-calibrated phylogenetic hypothesis, and we changed the number of components of how these analyses were run. So we changed the way the areas were coded for presence and absence, depending on the taxa of interest, and we changed the shape of the model. So we can use a time stratified model, so with different temporal layers, so close to the present, 20 million years up to today, 35 to 20 million years, 45 to 35 million years, 70 to 45 million years. So we create these layers, which are important for us to tell the analysis when the different continents and land masses in general, they were more or less connected. We don't know exactly how to do that. So we changed, that's letter B in the sensitivity chart on the left. We created different submodels of this connectivity between areas to see how much the probabilities of moving between areas could affect the overall results. And this, of course, if the results point to the same outcome, we can have more confidence that our analytical decisions are not making that much of a difference in terms of the overall conclusions we get. Talking about area coding for biogeographic analysis, look at the top taxon therein represented. That's Colletidae antipodis. So Colletidae is a small endemic taxon present in Australia and only in Australia. So when it shows it as being observed in Australia, I can say that all Colletidae occur in Australia because I know that's what we know about Colletidae. So the interpreted distribution of Colletidae is the same as the observed one for the species in hand. When we look at Colletidae, we have Colletidae almost everywhere. And of course, some Colletidae are known to occur in South America, in the Palearctic, in the Nearctic, and so on and so forth. But we can also simplify the area coding to show that all Colletidae and Colletidae in general should be interpreted as being from South America, because South America is understood based on results of different studies as having been the place where the early diversification of Colletidae took place. So we changed as the raw data from the raw data to a kind of treated distribution of data for each one of the taxa. And to my joy, when I saw that the results pointed to pretty much the same conclusions, I was really ecstatic because it was great. I was very anxious about the impact of making these decisions, which are justified in many ways, but can be debatable, can be criticized. I was a little bit weary, but when I saw that they converged, I was extremely happy to move on and say, okay, we're getting really solid answers to the questions we're asking. So now to the results of and what we got here. But before showing the integrated results, I should say a little bit more about the fossil record and how that was used to understand the paleo distribution of families. So if we think about Mellididae, there aren't that many fossils of Mellididae. In total, we had 221 fossils that were used to generate this paleo record of bee diversity through time. And here I show there's one reconstruction of where this one fossil of Mellididae from the Miocene is interpreted as having been located when the world looked like it was in the Miocene. I go back in time to the Eocene and I see this other record. It's from an amber fossil, and it's in a place that makes sense in an area that corresponds roughly to Europe nowadays. And the same here, we see another Eocene record with the world looking a bit different again, and the reconstruction of where this fossil was back when it was preserved there. So we did that again and again for the 221 fossils, and here I should acknowledge the importance of the very careful study or sub-study done by Diego Porto to map all these distributions and to reconstruct the exact placement of these fossils. And finally, we get to the overall results. And I'm going to show you this time layers. So this is the oldest time layer, 110 million years ago. And based on this picture, we can see there are two columns of rectangles. The one on the left shows what the reconstructions, the analytical reconstructions tell us about the distribution of the different bee families and different parts of the world. And the column on the right shows what the fossil record tells us. And in this case, in the early Cretaceous, the fossil record doesn't tell us anything. We don't have a bee fossil that can be attributed to the families we know nowadays. But the reconstructed distribution for the families show us very clearly, and I'm not going to show you the raw results at this point, and there wouldn't be enough time, but we see that all bee families are reconstructed to be in this supercontinent formed by Africa and South America before the breakup of these two large land masses. The one on the bottom, the red, as the legend shows you, is Mellididae. So Mellididae had already diverged from the remaining bee families, and so it was there. And the remaining ones, they have these little dots, meaning that that's the presumed distribution of the ancestor of each of the bee families, which had not differentiated yet. We move on to 110 to 90 million years ago. And we see that based on this result, and I think this is one of the coolest ones, that if my time machine took me to Africa, I would expect to find Apidae, shown in blue, Colletidae, shown in brown, and Mellididae, shown in red. And in South America, I would find Andrenidae, I would find Apidae as well. So Andrenidae is in South America, but it's not in Africa. Apidae is in both. Colletidae is in both. Colletidae is only in South America. And the ancestor of Colletidae plus Ctenotrididae would be in South America. And we move on to 90 to 70 million years ago. And now we have a consolidated picture of South America with Andrenidae, Apidae, Colletidae, Colletidae, Colletidae. We have Africa having Apidae, Colletidae, Colletidae, and Mellididae represented, and then we have Colletidae and Ctenotrididae already present in Australia. So it took a while for Australia to become the cool bee region we know today, but it was after the breakup between Africa and South America, but during the time when there was a connection between Australia, Antarctica, and South America, kind of confirming the results or the hypothesis proposed by Almeida et al. Some years ago. 50 to 70 million years ago, we have a different scenario. So here we start finding important evidence coming from the fossil record, and that's especially true for northern continents. And what we expect from this time when the planet was warmer is that bees that were originally in southern continents, they expanded their distribution to the northern continents, either from South America to North America or from Africa to Eurasia, and that had a profound impact on the later distribution of bee taxa around the world. And that's preserved on the fossil record, and it's better depicted by the fossil record than it is based on the analytical reconstructions. And as we move forward, the scenario is not going to change very much, but we see more and more signal coming from the analytical results up to the present, when we see a mix of what the DEC model reconstruction shows us with present day distribution, and a little bit of the fossil evidence also from the last 20, 30 million years. So we're getting there. I think the result is very promising, showing this two-stage history. First, an early diversification taking place exclusively in southern continents, later expanding to northern continents and allowing for a mixing of the faunas, which later on went southward to expand their distributions to areas where they were not present originally. And a lot of the detail present in these results will not be part of this talk. Otherwise, I would need another hour, and I'm not going to do that to you. Just to leave you with some key messages of a very strong biogeographic signal for the early diversification in Western Gondwana, Africa, and South America. The fossil record was essential to understand the northward range expansions during the Tertiary, particularly during the Eocene. And there are limitations in inferring the history of an ancient taxon like bees in a single analysis. So we get a lot of detail the way we've done when we look at the earliest stages of this biogeographic history, but we lose accuracy when we get closer to the present. And does our time machine work? I think so. I think what we've gotten from this approach, integrating different kinds of sources of data and different analytical methods, I think it gave us a picture of how bee biogeography can be understood that had not been achieved before. But we can also say that Charles Michener was right, as always. And I think that doesn't make our work less interesting or important, because a number of important research has reached the same conclusion. We've made a lot of progress, but in the end, Mitch was right, and which is good. I think it's always great to confirm what someone like Mitch already knew 40, 50, sometimes 80 years ago. So he was amazing, yes. And this talk, if it has to be dedicated to someone, it should be dedicated to Mitch. And with that, I would like to thank the co-authors involved in this work. It wouldn't have been possible to do this without help and this joint collaboration with Silas Bossert, who I emphasized before, but also Brian Danforth, Diego Porto, Bonnie Blamer, Felipe Freitas, Patricia Stroher, Marcel P., Elizabeth Murray, Michael Kuhlman, Michael Brandsteder, Lawrence Packer, and Sean Brady, and the funding provided by funding agencies in Brazil, in the US, particularly NSF, and elsewhere. So with that, I thank you very much for your attention, and my talk. We can move on to Q&A. Thank you.
All right. That was fabulous. Thanks so much, Eduardo. I really love those maps you shown at the end. What a great way to present the data, to summarize so much information over such a long period of time. 130 million years of evolution and biogeography in half a dozen slides. Spectacular. Okay, so there's only one question in the Q&A so far, and that is concerned about amber, Baltic and Burmese amber samples to get a more accurate picture of bee distribution. That comes from Josh Sato. So to rephrase it, what is the oldest amber? I actually don't know that.
Still is the Critotrigona prisca. It's funny, and it goes back to Michener again, 1988, I believe. Michener and Grimaldi published an important paper describing the stingless bee from New Jersey, which is not a particularly rich fossil deposit for bees. And after that, we have some compression fossils and ichnofossils from other parts of North America and South America. But the richest fossil records, especially thinking about amber fossils, are from the Baltic amber. We don't have any Burmese amber bee that we can confidently say this is a bee. We have lineages that are closely related to bees, others that may be closely related to bees, and there very interesting research being done with that. There is a postdoc working in my lab, Bruno Rosa, has been doing great work with that, and Gabriel Mello. So just to cite two people I know, but George Poinar has been very active, Michael Engel. So a number of people have been working very hard to tell us more about what was going on in the Cretaceous and the early Paleogene. And who knows, maybe a year from now, we're going to have a better or an older bee fossil from Burmese amber. Maybe not, maybe it was too wet for bees, who knows, or from other parts of the world with amber fossils. I don't know if I answered the question properly, but I hope I provided a number of facts that may be useful.
Well, that kind of leads me into a question that I had, and that is about the phylogenetic patterns associated with the outgroups that you're using and the biogeographic provenance of those. So as the classification of apoid wasps seems to indicate increasing fineness with respect to where the sister group of bees fits. You know, it was Cribronids until, I don't know, 10 years ago, and now it's Ammoplanidae or Ammoplanids, and maybe it's even just a subgroup of those. So how you know, I guess the question is twofold. One, you know, what was your closest outgroup to bees in the analysis, and what kind of phylogenetic pattern do you find among the outgroups?
Lawrence, the outgroup sampling, I avoided talking about it because it was not that relevant for the analysis per se. We focused the dating analysis and the biogeographic analysis on the in-group. There are 25 species of outgroup apoid wasp sampled for this analysis. And it's very similar. The sampling is very similar to that you find in Brandstatter et al. 2017, with a few additions which don't make that much of a difference in terms of the understanding of the close relatives of bees. So if one is interested in knowing more about the close relatives of bees, it's better to go to Manuela Zan's et al.'s paper published 2018, I believe. That's probably the most comprehensive sampling of the pamphredonines in the old sense, including what are the ammoplanids and other taxa. If the understanding of the biogeography of those apoid wasps is going to change our understanding of bee biogeography, I doubt it. I don't think it's going to change the picture I shared with you today. Because it's a very strong signal that comes from within bees. What we may learn, and we should learn as the phylogenetic studies of apoid wasps improve, is where the breakup between the closest relatives of bees and bees themselves happened. So is it something that also happened in Western Gondwana, or it's something that involves parts of what now are Eurasia or Laurasia and Gondwana, and the time frame for that is that early Cretaceous or it's late Jurassic. That will be really interesting to know, and why bees that may even help answering why bees are not found in the Burmese amber deposits, which are great for other kinds of insects, including apoid wasps. So. Yeah, if we think about biogeographic analysis as something that goes backward, so we're in the present and we look backward, it's great. We learn a lot about current distribution, and then we go back in time to try to estimate what ancient distributions looked like. It's also useful to go upward and see what close relatives tell us about this ancient distribution, and then match the two. So far, we've been doing mostly the downward pass, right? Go from the present to the past, and then learn a lot about that. But probably in a couple of years, we'll be able to do both and learn more about the setting of origin of bee, the origin of bees, and their closest relatives.
Okay, jolly good, thank you. From Catherine Odanaka, how are you able to pinpoint possible places of origin of bees and their groups, knowing that landforms were different at the suggested times of origin?
Yes, so when we talk about the origin of bees and we think about the actual location, we don't know for sure, Catherine. What we know is that as we go back in time, we see taxa that have a very strong and long-lasting association, historical association to one landmass or to two or to five. And then we see how the other groups that are related to that one, how their historical association to landmasses look like. And as we go back, we see that different groups are have been associated to either South America, Africa, or both, indicating that the area that included both of these areas, which is broadly known as Western Gondwana, was relevant. But if you were to ask me where exactly in Western Gondwana, why some groups are only in parts of this large landmass and others are apparently more widespread, that's harder to know because we're thinking more about the events, and the breakup of this large landmass was the key event here than about the areas themselves. The areas are kind of the proxies for us to think about the events, which are the key, and it's a bit vague, but that's what we have to work with. If we have a very rich fossil record for that time of bee evolution, then we could talk with more certainty about areas and not just about the general events and which areas are involved with those events.
Okay, super, thank you. Aline Martins, hi Eduardo, fantastic talk. Curious to see the paper. Oops, the question just got booted out. Okay, curious to see the paper published. Curious to see there was no mismatch between ancestral area reconstructions and fossil record among the geographic regions. Maybe this is an artifact of the analysis or just a nice coincidence showing the most probable history.
Yes. I think it's an artifact of the fossil record. It's could be defined as, in simpler terms, as very incomplete and gappy, but still extremely important to understand this expansion events that took place about 60 to 40 million years ago northward. So, as I mentioned, an analysis of this kind, especially if you're only thinking about the analytical models such as DEC, it's not going to be as reliable in every part of the time scale. It's going to give stronger results when we're closer the way we've done. It's going to give a much stronger signal towards the base of the tree. And the signal becomes blurrier as we're in the middle when the fossil record kicks in and gives us more information about what was happening. And then the level of detail we can expect from the analysis when we're closer to the present is really ridiculous. And that's why we used something that's not a family level analysis, just considering 7 lineages or 6 lineages or something like that. But we didn't use all the terminal species analyzed as species, because some of those species actually represent multiple taxa clumped together. So I mentioned Calomelyta antipodis. That represents the whole lineage of Calomelyta or Calomelytini. And so putting all that together, there are artifacts, and it's important to be aware of those when running the analysis, but especially when interpreting what the results are good for. yeah, hopefully I was able to answer your question. I went in different directions here and I kind of got lost.
Go on. Sophie Miller's got a related question, and that you mentioned that it is the case that the signal is stronger further back in time and more weak more recently. But why is that? What is that? Why, Sophie asked, why is it that you get? You could
okay.
say that.
I think it has to do with mostly with the refinement in terms of the taxon sampling you get for when you have an analysis that let's think about Andrenidae. I mentioned Andrenidae had these 2 amazing papers published recently, and so in those papers there were hundreds of species of Andrenidae. I don't know the numbers by heart, but there were more than 100 species of Andrenidae in each, and so every tribe and multiple species per tribe were represented, and sometimes you could have for a given tribe 4 different geographic regions well sampled and included in the biogeographic analysis. So that gives a level of detail that we're not able to have when there are only, let's say, 15 or 20 species of Andrenidae. We still get the ancient result because all the ancient signal, I should say, because all the major lineages that diverge in the first 0 to 20 or 30 million years of early Andrenidae evolution are represented. But then you start missing signal because you don't have as many taxa sampled. So we can learn more about the ancient divergences, but it's just an outcome of not having enough detail in the sampling of the in-group. So the following question could be, so the answer for that is to run an analysis with 20,000 species of bees. Is that what we should do? I don't think so. First, because if you have 20,000 species of bees, you're going to have a very hard analysis to be run. At least nowadays, it would be hard to do that. For a number of those species, you're not going to gain a lot of knowledge by just including every single species. And the other thing is, these analyses, they're always, I think, you're always going to have to make a compromise between the kinds of answers you're going to get. So it's important to keep an eye on the detailed analysis of Andrenidae, and then the overall analysis of bees, and then mentally bring everything together or to review everything in your mind. But instead of having one analysis that answers everything, I'm kind of skeptical that we should attempt to do a huge analysis that's going to solve all problems. I think it's better tackled if we do that by parts.
That's an interesting thought. Ruben Garrido asks whether it might be interesting to look for bees in areas where there are particularly ancient plant lineages.
I think that's a question for you, Lawrence.
Me?
Yeah.
I don't know anything about early plant lineages.
Oh, plant lineages. I'm sorry. I thought it was early bee lineages. Sorry. Yeah, no, because I pointed to Lawrence because Lawrence has been very successful in going to places like Chile and other parts of the world to sample lineages that may be important to understand the early evolution of some taxa. And in fact, he has found some groups that were undescribed that have been helpful to understand this early evolution of certain taxa. But early plant evolution, that's a harder one to answer. I think it will be important in the future to bring together explanations about early plant evolution and early bee evolution and see if there's a match. But in many cases, what we notice is that specific bee groups have radiated at the same time in probably in close connection with the radiation of certain groups of bees. And as a very nice example, Aline Martins, who just asked a question, has done that with the Centridini and found a very nice pattern of association temporally and in terms of the diversification of oil producing flowers and the bees that visit those flowers. So I don't know if the earliest flowering plants are going to necessarily be the key to understanding early bee evolution. I think the bees took advantage of an already evolving bee plant, already diverging flora, and then those bees were important for the flowers to radiate at the same time as the flower radiation was important for bee evolution. So yeah, maybe some groups of plants, but not necessarily the earliest ones are key for to answer the question you asked.
Well, I guess an interesting point with that is, Nilana and Melissa, which is a single species representing 70 million years plus of bee evolution, and yet the plant that it goes to, that genus is maybe only 5 million years old. So you mentioned Elaine, so she has another question regarding one of the interesting conclusions in Michigan 1979. Bees are more abundant and diverse in arid areas, and that they might have originated in such an environment. Does your data support that also? Like, so if you've got habitat, paleo-environmental data for Western Gondwana for 130 million years ago?
No, I haven't.
If not, why not?
That. Okay.
Sorry, say that again.
I haven't investigated that, and I'm not aware of anyone doing that at the moment. I think the hardest part about doing that is the kind of assumptions you do and the kind of analysis required to play with those assumptions. So it would probably be based on either a rich fossil record that would help us understand where fossil bees were when they first evolved, and to understand the kind of habitats that they were in. And the other one is to think about niche conservatism. So if you trust niche conservatism is going to be a good source of evidence to based on present day distribution of bee taxa, you go back in time and then see where those bees were, and then extrapolate that to times when we had no idea where bees were, but based on the biogeographic reconstructions, we say, okay, Western Gondwana land, what was going on there, and what kinds of habitats were in there. So it's feasible, but it's you're going to really have to bring together well-justified assumptions about niche conservatism and how it changes through time. And even the paleo reconstructions for some of these areas, for example, Western Gondwana land was a huge area. And in some parts, there were probably moist environments, and in others, arid habitats, where exactly bees were when they first evolved. That's harder to know without a rich fossil record, but maybe I'm just being too conservative in my thought about this problem.
So you got to discuss paleo environments with some dinosaur biologists.
Certainly not.
Yeah, I like your dismissing of reptiles and mammals as uninteresting parts of the fauna. And I'm just as biased as you are. Okay, so that's it for the questions from the audience. But I still have one. And actually, I have a bunch. But the one I want to ask in public concerns the interpretation of fossils. So did Diego look at most of these 200 odd plus fossils himself, or did he rely upon the interpretation of others?
The interpretation of others, yes. Diego actually didn't look at a single fossil for this analysis, but there were what was done was both this literature review based on the original descriptions and the interpretations that were published afterwards. So let's say Cockrell published, described the fossil in the 30s, and later on this was interpreted as a very dubious as having a very dubious taxonomy, or the fossil deposit was not trusted. Then some of the fossils available were discarded, and but yeah. So in short, it was a literature review in this case, and so we're going to have the database of these fossils made available with the actual names and the specifics of each of those fossils. But for the analysis integrating the paleo reconstruction and the analytical biogeographic reconstructions, we're going to have that done by family, so family by family. So it's one of the difficulties of dealing with the detail in different parts of the evolutionary strata. So we're going to have Apidae, which may be Anthrophoridae, but it may be Meliponini. Well, yeah, it's hard to explain without showing figures, but I think I made the point.
I got you. I look forward to seeing it.
Well, very soon. You have to comment on it very soon, Lawrence.
Oh yeah, I should hope I should have to comment, but that's fabulous. And like, but the issue is, of course, interpreting the fossils and even some of the more recent descriptions of fossils. When other people look at the same fossils, they come to different conclusions about the characteristics. The way I describe it to my undergrads is, it's difficult enough identifying something when you got a specimen in front of you that you killed earlier that same day. Now imagine squashing that insect between two rocks and leaving it there for 100 million years. It's really difficult to do that. So one has to have some sympathies with paleoentomologists, but none with paleovertebratologists, because they can see these massive fossils, and if they haven't got things sorted out from things the size of large buses, then there's no hope for the rest of us. All right, Eduardo, thank you so much. That was a great deal of fun.
Thank you, Lawrence. And thank you for everyone who's watching it. It was great. As I said, I would much rather be in a room with everyone and having the interaction, but it was great. And thank you for the questions too and the discussion in the end.
Well, you know what, to be honest, if you'd come to Toronto, I would have still watched the talk remotely. I've only been into the university on weekends since early March of 2020.
Wow.
And the only traveling I've done has been to go to France to see my daughter, son-in-law, and granddaughter. Not been on any field trip the entire time. Okay, so the last thing we're going to do is just to point out that there's a couple more talks that we've got scheduled. Actually, we're scheduled all the way through to the talk in October. The next one is going to be on the systematics, biogeographical history, and evolution of floral associations in userine bees. That's with Achig Dorchin and Felipe Freitas. From two different continents, so that's going to be fun. And the next one after that is in September, and I don't have control of the. Nora, can you put the next slide up, or do we not have it?
Yeah, sorry, no, that's all. We don't have another slide for September.
Oh, okay. Well, I can tell you that in September it's going to be collected. And that's going to be with Jason Gibbs and his student, whose name is completely irresponsibly slipped my mind. Joel Gardner. That's right. It came to me. I know I'd find it eventually. And if you found that incompetent, you wait till you're 67 and see how well your memory works. at short notice. Okay, so Joel Garden. Yes, Jess, I got it. Jason, thank you. So that's going to be at the end of September. Then in October, I think we go back to Belgium, and it's going to be Denny Michet giving a talk about European bee biogeography or something similar. All righty. Well, thanks everybody for coming. That was a lot of fun. You can, here I am, but I'm waving goodbye, and I'm receding into the distance, and my chair doesn't go any further back than that. So I will also hope to see you all in a month's time. And what a lot of fun that was. And I look forward to discussing this with Eduardo and others at length over the coming months. So thanks a lot for organizing this, Nora. You did a great job.
Thank you. Thank you for inviting me.
Well, I couldn't do it. Everybody knows how incompetent I am with social media and related technologies. I don't even have a cell phone. Okay, bye for now.
Well.
Thanks,
Bye-bye.
everybody.